Okay, so we're going to discuss about bacterial cell structure, physiology, metabolism, and genetics. So this just be the part 1 of that. Okay, so why are we studying here the bacteria or the microorganisms, so primarily the bacteria? So again, basically that one is we are classifying, isolating, identifying, and analyzing the bacteria. How are we going to isolate that one? So we have the specimen coming from your clinical sample and try to culture that one in the laboratory. Allowing the bacteria to grow in the laboratory would allow us then also to specifically identify the bacteria by either morphology identification or even the biochemical reactions. Okay, then we have here the taxonomy. So taxonomy is just a classification of your microorganisms or specifically your bacteria on the basis of their shared characteristics. So we have here the taxonomy, so just a hierarchical organization of your different organisms, including your bacteria. So we start here with the biggest organization. So we have here the domain. So the domain here could be classified as either your bacteria, archaea, another one is your eukarya. Followed here by the kingdom, and we have here the kingdom Animalia, Bacteria, Archaebacteria, we have your Protista, you have your Plantae, we have your Fungi, and many others. Then followed here by the phylum, then we have here the classification next to your phylum, we have the class, the order, the family, tribe. The family is actually pertains to the clan of your organism or the bacteria. And the genus, on the other hand, pertains here to the last name of your bacteria. And we have also here the species represent here the epithet or the first name, for example. So like, for example, if you have your Staphylococcus aureus, so the Staphylococcus here is actually the genus and it pertains to the parang last name nila. And the aureus here, the first name of that. This is the species or the epithet. Under that one, you could also have here some subspecies. Okay, with your species here, you could also have classify or group here the organism as to their strain. So example for that, we have your MRSA. So it's a type of Staphylococcus aureus characterized here as methicillin resistant. So that becomes the strain. On the subspecies level naman, we could classify or regroup that one according to the serovariety. Serovariety here is a classification based on the serological testing. So with the use of your reagent, so for you then to classify or group your microorganisms. Biovar, on the other hand, is based on that's your biovariety. Okay, the basis for that is based on their biochemical reaction is another test. Another one, you could have your genotype, we have also your phenotype, and other subspecies classification. Okay, then we have here the basis for the classification or the grouping here. So it could be in the form of your genotypic or phenotypic typing. So when speak about the genotypic here, genotype is based on the genetic characteristics or genetic makeup that primarily based on the sequencing of the nucleotides DNA or the RNA. Genotype here could be identified by your molecular tests, like for example, your PCR, polymerase chain reaction. Phenotype, on the other hand, pertains to the physical characteristics. Most likely, we could identify the phenotype of your bacteria by microscopic identification. Like you could perform here the gram stain, identifying that one as gram positive or a gram negative, or could also be identified through their shape, their size, or even their morphology or their arrangement. Or even biochemical reaction is also part of the phenotypic typing. Okay, now we go to the nomenclature or the method of naming or giving names to our bacteria or microorganisms. Okay, so if you're pertaining to the family, so always nag-end ng suffix na Aaceae, ganon. Like your Micrococcus, the family of that is your Micrococcaceae. Okay, so again, the method of giving names, scientific name here is a binomial system. So the first name is your genus. Okay, the second name is the species. For the genus, start with a capital letter and all the rest here are small letter, including your species or the epithet. Okay, when you're writing that one, so you need to italicize or underline. So always remember that when we are dealing with the scientific names. For the shortcut, like when you are writing, for example, your thesis, something like that, for the citation, you can shortcut that one on the second citation. Sabing second citation, the second time you try to cite or mention the scientific name. Um, you can have here the shortcut as either the first letter of the bacteria of the genus, and we have also here the species should be written here as a whole na species. Pero may mga bacteria kasi nga pareho ang kanilang first letter of their genus like your staph, your strep. Hindi tayo pwede maglagay ng S. aureus because again, pwede siyang strep, staph, so ganun. So for this bacteria, which has the same first letter of their genus, then you need to write that one as a shortcut. Like for example, your Staphylococcus here, you write your staph, period here, or strep. Okay, they follow here by the whole species name. Okay, on the other hand, sometimes you encounter here, then you try to write here Staphylococcus species. Pag nakalagay ang word na species here, mean to say, hindi naka-identify kung anong klaseng species of your bacteria. Okay, and remember na walang plural ang ating species. Pag isa man siya or madami, still species pa din siya. Walang species na word actually. So for the shortcut of your species here, so SP, pag singular, SPP for the plural. Okay, so that one. So pag sinabi na strep species here, streptococcus species, so hindi natin ma-identify o hindi na-identify kung anong specific na species for that. On the other hand, you could also write the group here like Staphylococci or Streptococci. Pag group siya, this one is not a capital letter, hindi siya naka-underline, at hindi rin siya naka-italicize. Again, this is a group. This is not the same as your family na naka-end siya sa Aaceae. Ganun. Okay, so other bacteria would have here the common name. It's also being called here their other names. You'll be encountering much of the other names when we discuss each of that different bacteria. So example for that, we have here the gonococcus. So this is our parang universal language. We speak about the gonococcus that pertains to your Neisseria gonorrhoeae. Pneumococcus is your strep pneumoniae, streptococcus pneumoniae. Tubercle bacilli is your Mycobacterium tuberculosis. Typhoid bacilli, we have here your Salmonella typhosa. And we have here the Friedlander's bacillus, or also called your Klebsiella pneumoniae. Okay, then we have here the difference between the structure of your prokaryotic organisms and eukaryotic organisms. So most likely, ang ating prokaryotic organisms is only your bacteria and your archaebacteria. Archaebacteria are mga bacteria which try to thrive at an extreme temperature. So sila yung mga thermophiles, so they're living in high temperature. Sila yung mga halophiles who try to live on the high salt environment. That would differentiate that one from the usual na mga bacteria. But then again, they're still part of the prokaryotes. So the difference between your prokaryotes and your eukaryotes here. So, okay, the first one, we have here the membrane. We have here your nucleus. So, nucleus here could eventually be found only in the eukaryotes. Walang nucleus ang ating prokaryotes. For their chromosomal DNA, since may nucleus ang ating eukaryotes, so their chromosome here could be found inside their nucleus. For the prokaryotes, since they don't have here the nucleus, they still have here the chromosome, only that they are found in the nucleoid or a sac-like structure but that one is not found in the nucleus kasi wala siyang nucleus, but rather that it's being suspended in the cytoplasm most likely. For the organelles, so eukaryotes here would always have that one, whereas prokaryote would have no organelles. For the cell wall of your eukaryotes here is made up of the polysaccharide, and for the prokaryotes again, that one is made up of the peptidoglycan. Cell structure, appendages, or extension. So makikita mo yung mga extension ng kanyang cell membrane. So like in the case of your pili and your fimbriae, that could only be found here among the prokaryotic organisms or bacteria, but not in your eukaryotic organisms. Okay, now we go to the different structure here found in the prokaryotic prokaryotic cell. So we start here in the cytoplasm. So ano makikita natin sa cytoplasm ng prokaryotes natin? The first one, we have here the nucleoid or the nuclear area. Since again walang nucleus ang ating prokaryotes, so you could have here the nucleoid found in the cytoplasms, not within the nucleus. So this is serving as its chromosomal DNA, and we describe the chromosomal DNA here as a single mag-isa saka circular chromosomal body. And usually that one is attached in the sac-like structure. You call this one as your mesosomes. Another one, we could also find plasmid in the cytoplasm of your prokaryotic cell. The plasmid is another extra chromosomal DNA. It's a circular, double-stranded DNA, but this one is also circular. And this plasmid is responsible here for conferring resistance to several antibiotic of your bacteria. Itong plasmid na to is being exchanged by your bacteria from one bacteria to another that would render them, since they are exchanging their chromosomal DNA, that would allow them to confer resistance to antibiotics, which is normally dapat hindi sila resistant, but they're able to acquire this plasmid here coming from another bacteria through their genetic exchange, and therefore that would render them as well here to become resistant for that antibiotics. Another one, you could also find here inclusion in the cytoplasm of some bacteria. Example of the inclusion, we have here the granules. So the granules at the cytoplasm is usually serving here as the storage granules. They serve as a storage here of, number one, pwede siyang storage of their polysaccharide in the form of the glycogen. Pwede din magiging storage granules ito made up of the lipids, primarily. Specifically, we have here the poly-beta hydroxybutyrate, and we have also the polyphosphate. In the case, for example, of your Corynebacterium diphtheriae, that bacteria contains the specific na granules. You call that one as the metachromatic granules. The metachromatic granules of your Corynebacterium diphtheriae is actually made up of the polyphosphate. So the serving here as its storage form of the phosphate for its ATP production. Another one, some bacteria contains here the spores. So the spores here, so we have two form of the spores. One is the endospore. We consider here as the dormant, inactive, and asexual spore. Kasi yung mga fungi natin, gumagamit sila ng spore nila for them to multiply or reproduce. But then again, this is asexual. Hindi ito for reproduction. On the other hand, we have here the so-called vegetative form or the vegetative spores. This is the active spore. The endospore become vegetative spore once the bacteria has been exposed to the harsh environment. What are examples of the harsh environment? Again, they serve only here as the protection of the bacteria for them to withstand the harsh environment. Like what? Like the presence of the chemical agents, reagents. We have also here dehydration, desiccation, drying up. We have also here the high temperature, and even the presence of your gamma and your ultraviolet radiation. Okay, the endospore here, the spores could be identified in the bacteria if you try to have here the gram stain. For gram stain, they are colorless area at the center. So, makikita mo parang may space that become the spores. That's actually the spore. And we have your specific na stain for the spores. We have the Shaver Fulton. That would have your better picture of your spores if you try to use a stain. Pero pag gram stain kasi, magiging ano lang siya, colorless. It will appear as a colorless area at the center of the cell. Okay, we classify also here the spores according to the location where you could find that one within the cell. So, pag nakita siya sa gitna, that's the central. So, subterminal here, and this is your terminal. So, para sang drumstick or lollipop. So, become here the terminal spores. Okay, so we have here the two genus of your bacteria having the spores. We have the genus here, Bacillus, and we have also here the Clostridium. Only that the Bacillus bacteria, the genus Bacillus here, this includes Bacillus anthracis, Bacillus cereus, and many others. And they are all, Bacillus are gram-positive. Your Clostridium na genus, on the other hand, like Clostridium tetani, Clostridium botulinum difficile, for example, those are gram-negative na bacteria na may spores. So, after the cytoplasm here, papalabas na tayo. Okay, so, if this one is your cell, so dito na tayo sa kanyang outer membrane. Okay, so we have here the cell envelope here that try to surround your cytoplasm. We start here with the PM or the plasma membrane. Okay, the plasma membrane of your bacterial cell here is primarily made up of the phospholipids with the embedded na protein. So basic naman yan, di ba? Phospholipid ang ating cell membrane, always a phospholipids with the proteins embedded on that. Nasa gitna may mga proteins. For the prokaryotic organisms, so basically that one, it's made up of the phospholipids plus the proteins minus the sterol. Sterol here is a cholesterol, except your mycoplasma. Mycoplasma is the bacteria na meron silang sterol. All the rest of bacteria, wala silang sterol. So, mycoplasma is an exception for that na mga prokaryotic or bacteria natin, but because they contain here the sterol. Usually, the sterol is the main component of the plasma membrane of your eukaryotic organisms. For the function of your plasma membrane, so provide here as an osmotic barrier against the osmotic pressure. Another one is the site of your electron transport chain where they try to produce energy or ATP. Okay, the next one, after your cell membrane or the plasma membrane, we have here another coating. So, kung ito ang cell natin, so may cell membrane tayo, and another layer for that become the cell wall. Other name the cell wall is your peptidoglycan or the murin layer. So, basically, the function of that is possible here for providing shape, maintaining the shape of your cell. And basically, that's to provide here to maintain the rigidity of the cell. And another function, try to protect here the cell against the osmotic gradient or the osmotic pressure to prevent bursting of your cell. So, what would be the component of your peptidoglycan? So, peptidoglycan is a very complex structure primarily made up of the polysaccharide. Pag sabing polysaccharide is a sugar. So, for example, this is your polysaccharide. So, may sugar dito. In each of this one, may alternating na layer of your NAM and NAG. NAM is your N-acetyl muramic acid, N-acetyl glucosamine. So, alternating, we have here the NAM, NAG, NAM, NAG, and NAM, NAG, and so on. Another thing, ano pa makikita natin sa structure? In the NAM layer or N-acetyl muramic acid, may naka-attach na 4 na amino acid. You call that one your tetrapeptide. That includes your L-alanine, D-glutamate, mesodiaminopylimate, and we have also your D-alanine. So, one polysaccharide unit, we're able to bind to the next polysaccharide unit by this amino acid na tetrapeptide. So, if, for example, you have your NAM, may 4 dito na tetrapeptide. Another layer of your unit of your polysaccharide, ang NAM area niya may tetrapeptide din. By the tetrapeptide, another tetrapeptide here, they able to form here a linkage by the peptide bond para mag-form siya ng bigger structure comprising approximately here the entire peptidoglycan unit.